So, let us resume where we left in the last class. We are talking about the non-enzymatic glucose sensor. So far we talked about 3 technologies, and let me highlight one thing. I am only highlighting the major papers, but that by no means that we are neglecting the other ones. It is just the time is a constraint. So, I cannot explain all the possible materials what people have used. These are some of them, but no offense that there are other studies too, and because of the exhaustive nature of the course, I am unable to cover all of that, but the basic principle is this. So, we talked about the first one about copper oxide, zinc oxide hybrid material as a electrocatalyst. Second, we talk about copper oxide decorated on top of laser induced graphene, second technique. The third one we talked about the nanograss, the grass like structure protruding out from the surface of copper hydroxide, right. These are the 3 we have talked. Now, we will talk about the fourth one in that series. So, the fourth one is copper foam for glucose detection. This is our title of this class to start off with. So, the miniaturization of the copper foam nanodendrites and their use for glucose electrooxidation, which is figure D. These authors firstly fabricated 2 different band gold electrode. Why we use mostly the gold electrode? Of course, it is a very costly thing though, but it is reasonably good in terms of inertness, and secondly conductivity is fairly high and you get a noise free recording with gold or platinum electrodes. That is the reason why you use the gold electrodes. Okay. Unless we move to a commercialization level where we can replace it with other economical electrode, and mostly the focus is currently across the world on carbon based electrode systems, which are the most economical among all of them. So, gold electrode arrays at micro scale, and this was followed by the electrode deposition of copper foam. So, you have say for example, gold electrode lying there, on top of that you have these foams of copper foams. By applying negative high voltage on in an acidic solution in the presence of Cu2 plus ions. The study explains the surface composition changes of copper oxide species before and after glucose oxidation process. Furthermore, the sensor exhibited superior analytical performance with a sensitivity of an outstanding sensitivity of 10.6 microampere towards glucose, while wide linear range up to 22.5 millimolar. So, if we look at the picture of this, this is the picture. So, what you see underneath is the electrode deposition. Prior to that, what you see is the gold electrode. On the gold electrode, you grow the copper, and this is where the reaction is happening. So, this is the most basic of these kind of electrodes. So, these 4 what we talked about, which started with Cu1 ZNO hybrid, then copper nanoparticle, and when you talk about laser induced graphene and copper nanoparticle LIG hybrid, then we talk about copper hydroxide, and now we talk about copper foam on gold. So, these are the 4 examples of non-enzymatic methods of detection of glucose, where you can bypass the glucose oxidase. Now, another very promising area which is slowly getting populated with literature and technologies is the wearable non-invasive electrochemical glucose sensor. So, now people want, like, you put on a wristwatch, you want it should be able to tell you what is the glucose level of your body, or just like a thermometer, you put a thermometer under your arms and it tells you the temperature. So, you should be in a position that you put something under your arms based on the sweat. It should be able to predict that how much glucose is there in your blood. So, these are all non-invasive methods. Or you put a contact lens, it should be able to tell you how much glucose is there in your tears. So, some of these non-invasive methods prefer to use sweat, tears. Some of these are fluid which are on our body surface, skin temperature, skin secretions other than sweat or in addition to sweat. So, these are what acts as analyte. So far we talked about, if you look at it, we talk about blood glucose or glucose in urine, right. Now, we are talking about situations where glucose level is lower and far more challenging than having it deriving it directly from the blood or from the urine. And this is the future, non-invasive sensors, because this is what the whole biodefense detection world is looking towards. Non-invasive methods, methods which do not need to draw blood or do not need to draw saliva, or even sometimes you can consider saliva also, just or tears, skin temperature, skin secretion, interstitial fluid. Of course, those are invasive ones though. So, what are the challenges? The majority of detection technologies for determination of glucose concentration rely on blood or serum analysis. It is also possible to detect glucose from other bodily fluids such as sweat, saliva and tears. For this purpose in particular, non-invasive glucose sensing platforms are of great interest, since they may be the ideal candidate for diabetes management. Such sensors do not come in contact with the blood, thus they are not exposed to the immune system, as happens in implantable glucose sensors. So, this is another aspect what you have to realize whenever you are taking urine sample, blood sample, there are cellular components which are big block, because we are talking about something in the range of 30 microns, which is the size or the diameter of a cell, a mammalian cell. So, whenever you are trying to draw the blood, it contains immune cells like macrophages, okay, and all these are acting as barrier for the detection and thereby bringing down the efficiency of your detection. And you have to realize the same problem comes when you are detecting virus, same problem comes when you are detecting microbes. They all are extremely heterogeneous mixture, And that is why I am taking so much time to get you the feel that how challenging the field becomes, because there are lot of interfering molecules. So, now however, the major problem related to wearable system arise from the physiological nature of external body fluid such as tears, saliva, sweat, and due to the challenges of reproducible sample collection. There are a lot of things happening in our body. Say for example, the most simplest thing, if you think of it, we use deodorants, all of us uses it. We use perfumes, all of us uses it. We use different kind of lotions, creams, different kind of skin improvement compounds. They all alter our chemical composition of tears, sweat, and other external body fluids. So, that is why the challenge is the reproducibility and how to get rid of those, because how to distinguish that that particular compound is not coming from the body, it has been applied on the body, and that is why you are seeing it. So, you realize that the challenge is far more complex than we previously even thought about. So, one of the ways they are trying to do is called a skin tattoo. What they do is that the system used a low current density of extract the skin interstitial fluid to decrease the skin irritation, and selective sensing of glucose by using glucose oxidase modified Prussian blue transducer was achieved at low applied potential. The authors also examined that the body performance of the tattoo based ionophoretic biosensing platform and to compare the changes in the glucose level pre and post meal. So, this is this tattoo thing, tattoo based non-invasive glucose sensing platform, schematic image of the printable ionophoretic sensing system, which is out here. The photograph of the tattoo based glucose sensor device applied to human object. So, you are seeing this is where the tattoo is being made, applied to human object. 3, the schematic image of the time frame of typical on a body study. This is time frame we are talking about. So, this is the one of the non-invasive methods. So, where you are basically using Prussian blue and seeing the change, but realize one thing, all these processes. So, again here what you are seeing is it is an enzymatic assay system, which is utilizing the just the surface layer of fluid in your body for glucose detection. This is one of the methods which are being used. There is a second method, what you see is the microfluidic device design and operation. So, this particular design is based on a flexible epidermal microfluidic detection platform fabricated by integration of lithography and screen printing technologies in order to achieve effective sweat sampling for continuous glucose and lactate monitoring. So, when we are talking about sweat sampling, this could be used for virus and other microbial detection too. So, most of the world of sensor biosensors have revolved around glucose, because that is the benchmark from where people move to other kind of sensors. That is why I am focusing exclusively in a big way for these glucose biosensors. So, this is that second kind of sensors we talk about, microfluidic based, where schematic image of the layered microfluidic device configuration, which is out here. The schematic representation of the microfluidic device, sweat collection and operation on skin. Here, photograph of the device integrated with wireless conformational electronics on skin. So, this was developed in University of San Diego, and that is why it is that logo says that UC San Diego. Okay. This is the second level. The third one, which is the C, what you see is the contact lens sensors fabrication process and resulting contact lens with embedded sensors. So, this is basically you are talking about now tears. So, if this is your eye and this is the lens of the eye and this is where the retina is. So, the contact lens is put here. So, contact lens is coming in touch with tears. So, if you have a sensor on that contact lens, then you technically can detect the amount of glucose present in the tears. So, this third one is fabrication and use of contact lens. This is figure C with the embedded sensors for glucose measurement from tears. For the construction of the contact lens, a PET polymer is used as a substrate and spin coated with a resist. This is followed by the metal deposition and lift off in acetone. The fabricated electrode are cut into small pieces with 1 centimeter diameter and then heat molded to shape of a contact lens. So, the contact lens is almost like an electrode. To achieve the immobilization of the glucose oxidase enzyme, a solution of enzyme is drop casted on the electrode surface and then the surface is suspended vertically above the titanium isopropoxide solution in a sealed dish to create glucose oxidase titania sol gel membrane. After forming sol gel membrane, surface is covered with Nafion. Once again the use of Nafion is pretty extensive. Developed non-invasive glucose monitoring system is studied with amperometry. Such sample micro-sized glucose sensors fabricated on a polymer contact lens showed a good sensitivity of 240 microampere. However, many characteristics remain to be improved, such as stability, full biocompatibility for wearable contact lens, integration with readout communication circuit, and of course, integrating into the wireless system, so that one can directly get a recording. So, here you have the whole process out here, where this is the electrode configuration, this is the reference electrode, and this is how it really ready for being put as a contact lens. So, if you look at it where we started, now summarizing it, we started with diabetes, we started with insulin as an agent of which could be used for killing somebody. Then we talked about diabetes, then we talked about how we can detect glucose in the body. From there we moved on to the basic reaction. From the basic reaction of glucose plus oxygen in the presence of glucose oxidase leading to the formation of gluconolactone plus peroxide, and then we move to the Clark's electrode, which is the oxygen electrode, detection of oxygen and amount of oxygen getting consumed in the process of oxidation of glucose became the benchmark of detection of glucose. Further, this was improvised where on the electrode surface where hydrogen peroxide is being formed electrocatalytically, it was oxidized, regenerating the oxygen, but in that process ejecting out 2 protons and 2 electrons, and the amount of peroxide became a direct measure for the amount of glucose which initially got oxidized in the process. Thereafter, we talked about utilizing this basic reaction to develop the wide array of glucose sensor, all the enzymatic arrays. We went through a series of them. Thereafter, we realized that how these complex fluids in our body, including serum, urine, sweat, tears are posing problem for enzymatic detection because of interfering molecules and inability of the reactant, in specifically when we are talking about the glucose to reach the glucose oxidase or the enzyme surface and entering its pocket. So, thereafter we talked about the advancement in the non-enzymatic detection. The last class we talked in depth, and in this class we talked about it. Thereafter, we moved on to the non-invasive methods, where we talked about how we can implant such devices on the surface of the skin using a tattoo, or we can implant them in a contact lens. And I am pretty sure in the years to come, we will have all these kind of surface detection methodologies, where we will be implanting them in a non-invasive manner on the skin surface. So, from here, in the next class, we will summarize all the different technologies which have developed for glucose sensing so far. There are far more technologies, and why you people need to know this, because that is the basis, that is the premise on which most of the developments are happening in the world of biosensing, because this is the benchmark. If you can do this one, everybody believes, then you can do the other ones, because this is well documented in the literature. Further, one more thing I wish to add, the way nanotechnology is getting inroads into the area of biosensing, and how different kind of nanomaterial like copper oxide, zinc oxide sandwiches, copper oxide graphene hybrids, copper hydroxide, cobalt, all these different kind of nanomaterials which are potentially powerful candidate in electrocatalysis are gaining tremendous popularity for non-enzymatic methods, along with different kind of flexible material for flexible biosensors, and apart from it, different kind of carbon based electrode materials, which are economical alternative to the gold electrodes, and always the troublesome ones are the copper and the aluminum, which gets oxidized very fast. So, there is immense popularity with carbon based like carbon quantum dots, graphene oxide, so graphene based systems. So, we are entering into a very interesting time when electronics is truly getting married with biological systems in the area of detection, therapy, and surveillance. So, I will close in, and the next class will summarize, will try to summarize in one go all the different glucose measurement systems, because that will pretty much will be revision of all the different technologies what we have studied so far. Thank you.